Hi everyone. In this lecture, we'll begin with transcription in bacteria, that is prokaryotes. Bacterial transcription can be divided into three stages. Initiation, that's how the transcription begins. Elongation, and then termination. In this video, we are going to learn or talk mostly about initiation of transcription in bacteria. A bacterial gene may be divided into three sequences with respect to its transcription. So what is a gene? How would you define a gene? So a gene is a sequence of DNA which is transcribed to mRNA, that is transcription process, and then the mRNA is translated to a protein or a polypeptide. That's central dogma. So that's the definition of a gene. And in context to transcription, a gene may be divided into three parts. First is the promoter sequence, then the RNA coding sequence, which is shown in red here. This RNA coding sequence is the middle part, is the sequence which is copied into mRNA, and the last is the terminator sequence. So this coding sequence begins from here, where the +1 is started. And as I said, this coding sequence is the sequence copied into mRNA. So +1 is the first nucleotide to be copied into mRNA. And this coding sequence becomes one part of the gene. Last one is terminator. So what is the promoter sequence? The sequence before the coding sequence. This promoter sequence is upstream of the RNA coding sequence. In genetics, the technical word is upstream and downstream. So +1 is the first nucleotide to be copied into mRNA. All the nucleotides before +1 called as upstream, and all the nucleotides after +1 are called as downstream. So promoter is the sequence which is present upstream of the RNA coding sequence. So what is the role of a promoter? Now, it's the same diagram, but one more thing has come here. This is the RNA polymerase enzyme, which is DNA-dependent RNA polymerase, and which we have seen in the previous lecture. This RNA polymerase is the enzyme which is going to do the transcription. And this RNA polymerase first binds at the promoter sequence. It orients itself at the promoter sequence, and by doing that, the enzyme comes to know in which direction it has to move. For example, in this case, in this direction to synthesize the mRNA. Which one is the template strand, which will be copied into mRNA? Like in the previous lecture, we have said that since DNA is anti-parallel in nature, one strand is 3 to 5, other strand runs in 5 to 3 direction. The template strand is the strand with the polarity 3' to 5', because mRNA is synthesized in 5 to 3 direction. So the RNA polymerase, by binding to the promoter and the way it interacts with the promoter, comes to know in which direction it has to move, which is the template strand, and where exactly to begin the transcription. Now, when we say where exactly to begin the transcription, like from where to start synthesizing the mRNA, which is the first nucleotide that will be copied into mRNA, and that is the +1, the transcription initiation site. So +1 is the first nucleotide copied in the mRNA, and is shown +1 is from where this red color part begins, that is the coding sequence begins. The region before that, which is the promoter sequence, is not copied into mRNA. As we said, that +1 is the first nucleotide to be copied into mRNA, and the nucleotides before +1 we say are upstream of that, and the nucleotides after +1 we say are downstream of that. We have written here U and D. So the first nucleotide transcribed is numbered +1. All the nucleotides downstream of that are assigned positive numbers, as you can see here, and all the nucleotides upstream of +1 are assigned negative numbers. So -35 means 35th nucleotide before +1, and +35 means 35 nucleotides after +1, or let's say +85 means 85 nucleotides after +1. So the promoter sequence always have negative numbers. There are two DNA sequences in most promoters of E. coli genes. They have been shown critical for specifying the initiation of transcription. So we are learning initiation of transcription, how transcription is initiated in bacteria. So with E. coli as our representative, what scientists have discovered that in every gene has a promoter. So in E. coli genes, most of the promoters have two DNA sequences, and these two DNA sequences are critical and they play a very important role in the initiation of transcription. And these sequences are found at -35 and -10. When we say -35 and -10 means 35 and 10 nucleotides upstream or before +1. So these nucleotides are in the promoter, and that's why they have negative numbers. These sequences, which are found at -35 and -10, are critical for specifying the initiation of transcription. The consensus sequence for the -35 region is TTGACA, also called as 5' to 3' TT-GACA. And the consensus sequence for the -10 region, also called as -10 box, formerly called as Pribnow box after David Pribnow, the researcher who discovered it, is TATAAT. It's also called as 5' to 3' TATA-AT. So when we say that these two sequences are critical for specifying the initiation of transcription, and they are found in the promoter sequence. So this sequence is found at the -35, that is 35 nucleotides before +1. This is found at -10, 10 nucleotides before +1. But when we say -35 and -10, we are talking about a single nucleotide. And this sequence is about, let's say, 6 nucleotides. This is also of 6 nucleotides. So, which means that -35 means the 35th nucleotide can fall anywhere here. And similarly, -10, the 10th nucleotide before +1 can fall anywhere. Like this can be -10, or this can be -10, so on. And these two sequences are found in most of the promoters of the E. coli genes. So these are consensus sequence. Now let's define, or let's learn more about what is a consensus sequence. Because these sites are similar in all promoters, they are called as consensus sequences. The examination of many promoters in E. coli, and not just E. coli, even other bacteria, reveals a general feature. Most of the nucleotides in the promoters vary in sequence from one bacteria to another, and also in the same bacterium. But there are short stretches of nucleotides which are common, and they are found in most of the promoters. And these short stretches of nucleotides are these -35 TT-GACA and -10 TATA-AT. The spacing and location of these nucleotides relative to the transcription start site are similar in most promoters. These short stretches are called as consensus sequences. This is an example here. They've shown 4 prokaryotic genes, or I shall say more specifically promoters of the 4 prokaryotic or bacterial genes. And in these 4 promoters of bacterial genes, they're talking about the -10 consensus sequence TATA-AT. So it doesn't mean that all the promoters of E. coli genes have exactly this TATA-AT sequence. This has been derived. Like for example, in the 4 genes you see, 3 of them have thymine at this position. So thymine is very common. So first nucleotide becomes T. Here also out of 4, 3 has adenine. Next also out of 4, 3 genes have thymine. Next also 3 genes have adenine, and so on. So this is the commonly derived consensus sequence, and this is how they have derived the -35 and -10 consensus sequence. Now, let's go more into the initiation of transcription, and for that we need to learn the RNA polymerase enzyme, structure and function of the RNA polymerase enzyme of bacteria. Only one type of RNA polymerase is found in bacteria, which is DNA-dependent RNA polymerase. So all the genes, whether they are protein coding genes, which gives rise to mRNA, which gets translated into protein, or whether they are tRNA or rRNA genes, all genes are transcribed by this one RNA polymerase. Yes, there are genes for tRNA, which code for tRNA transfer RNA, which carry amino acids during the translation process. There are genes which code for ribosomal RNA, which becomes part of the ribosomes. But this tRNA and rRNA doesn't get translated. As we know, only mRNA gets translated. But yes, there are genes that code for transfer RNA and ribosomal RNA. Before we go into the structure and function of the E. coli RNA polymerase, let's learn a very basic concept. So this, what you see on your screen, the different colors used. This is the first row, I can say, is the DNA or the gene. This is the messenger RNA, and this is the polypeptide or the protein. There are certain proteins which consist of single polypeptide or one subunit. So for such proteins, there is one gene. That gene gets transcribed into an mRNA, which gets translated into the polypeptide. And that only is the protein, because that protein is made up of a single polypeptide or subunit. But in contrast, there are proteins which are made up of multiple polypeptides, or called as multi-subunit proteins. Now, how these multi-subunit proteins are made? So each polypeptide or each subunit of that multi-subunit protein is coded by a separate or individual gene. So in this case, let's say this is gene 1. This, when transcribed, synthesizes this mRNA, which gets translated into polypeptide. And this is second gene, which is shown in blue, which when transcribed gives rise to mRNA, which gets translated into the blue polypeptide. And there's third gene, which is shown in green, fourth gene, which is shown in purple. These, when transcribed, give rise to their respective mRNAs, and which translated into the green and the purple polypeptide. And same is the case for the yellow one. So after you have these polypeptides made, these polypeptides are gonna come together and associate with each other to form that multi-subunit enzyme or protein. So let's say this red polypeptide, or shown in red, comes, and the polypeptide shown in blue associates with it. Then the polypeptide shown in green also associates. The polypeptide shown in purple also comes here and binds, and the polypeptide shown in yellow color also comes and binds, and this becomes the multi-subunit protein or multi-subunit enzyme. And for this, the hypothesis, one gene, one protein was revised to one gene, one polypeptide, that the gene codes for polypeptide and not the protein, because there are certain proteins which are more than one polypeptide or multi-subunit, as shown here, and there is a separate gene for each of those polypeptides. Now, why did we revise this? Because the DNA-dependent RNA polymerase enzyme of bacteria, let's say E. coli, the transcription enzyme, is a multi-subunit enzyme. It has many subunits or polypeptides, and for initiation, the enzyme which is involved is the RNA polymerase holoenzyme. Holoenzyme is a word not just for a multi-subunit enzyme, which it is, but also complete enzyme. So for initiation of transcription, the enzyme involved or required is RNA polymerase or DNA-dependent RNA polymerase holoenzyme, complete enzyme. What is holoenzyme? Holoenzyme is core enzyme plus the sigma factor. Next question comes is, what is core enzyme? The core RNA polymerase of E. coli consists of 2 alpha subunits, which you can see. There's an alpha polypeptide, 2 copies of it, 1 beta subunit, 1 beta prime, and 1 omega. So these 5 polypeptides, alpha, alpha, beta, beta prime, and omega, makes the core RNA polymerase of E. coli. And when we say 5 polypeptides, there are 5 copies, but there are 4 types of polypeptides, alpha, beta, beta prime, and omega, because alpha is in 2 copies. So like I said in the just a discussion few minutes back, that there will be a gene when transcribed and translated gives rise to the alpha polypeptide. There will be a separate gene that codes for beta prime polypeptide. There's a separate gene that codes for beta polypeptide, and there's a separate gene that codes for omega polypeptide. After translation, all these polypeptides associate with each other to form the multi-subunit protein. So to come again, alpha, alpha, beta, beta prime, and omega forms the core RNA polymerase. This is the RNA polymerase core enzyme. One more polypeptide, which has obviously its own gene, and when translated, transcribed and translated, sigma factor or sigma polypeptide, when it's added to this core enzyme, becomes the RNA polymerase holoenzyme. So when I said that for initiation of transcription, the enzyme involved or required is RNA polymerase holoenzyme, and RNA polymerase holoenzyme is core enzyme plus sigma factor, which we have seen. The moment you add sigma to the core enzyme, you get a holoenzyme. So why is that the RNA polymerase holoenzyme is required for initiation? If holoenzyme is not there, can core enzyme do the initiation? No. So which means the difference between core and holo is this polypeptide sigma. This is an important polypeptide for initiation, and yes, we'll see in the next video how this sigma factor recognizes the -35 and -10 consensus sequence, and it's a key player in initiating the transcription in bacteria. So without sigma factor, can the core enzyme do the transcription? The answer is yes. Can it make the mRNA? The answer is yes. Then if it can do the transcription without sigma, the core enzyme, and can make the mRNA, then what is the problem? It can synthesize the mRNA from anywhere, from any position. And it can make a mRNA which will be mostly non-functional or erroneous, incorrect, because the core enzyme can do the transcription, can do the denaturation of DNA, can make the mRNA, but that would be incorrect because the core enzyme doesn't know where to sit, where to bind on the DNA. And by that, the core enzyme will make an incorrect mRNA. So sigma factor helps the enzyme in recognizing the promoter sequence, recognizing the -10 and -35 consensus sequences, and helps the enzyme in doing the transcription at the correct site and every time when the gene is transcribed. So without sigma, the core enzyme is blindfolded. So this is the promoter, as we have seen earlier, where the RNA polymerase binds. It's an holoenzyme and it has the sigma factor. The sigma factor ensures that this enzyme binds at the promoter, and by binding at the promoter, it contacts the -35 and -10 consensus sequences. And by binding at the promoter, the enzyme knows in which direction it has to move, which is the template strand, which it has to copy, and where is the exact transcription initiation site, which is +1. And sigma factor reduces the non-specific binding of the enzyme and ensures that the enzyme binds only at the promoter and does the transcription from the correct site. And as you know, the interesting fact is that, as we said, that the sigma factor is a key player in initiation and helps the enzyme in recognizing the promoter sequences. Sigma factor is not required for the elongation and termination stages of transcription in bacteria. And in fact, in the next video, we'll see as the transcription begins and little bit of mRNA synthesized, the sigma factor dissociates from the enzyme, and the rest of the transcription is carried out by the core enzyme alone, which proves that sigma factor is involved or required in the initiation of transcription. So in this video, we have begun with the transcription in bacteria. Initiation of transcription in bacteria. We have seen how a gene is divided into three parts: promoter, RNA coding sequence, terminator. We have seen the significance of promoter sequence. We have seen the consensus sequence, -35, -10. What is consensus sequence? What is its significance? And how certain enzymes are multi-subunit enzymes. Then the RNA polymerase enzyme of E. coli, difference between core RNA polymerase and RNA polymerase holoenzyme, its structure, its subunits, function, role of sigma factor.